Infectious disease is one of the greatest health challenges in the US and in the world. And to combat viral and bacterial infections, we need new tools. My name is Mohammad R, Seyedsayamdost, and I'm a biological chemist. Our research is motivated by both fundamental questions and application-based ones. On the fundamental side, we're interested in uncovering and understanding the small molecules with which bacteria interact with their environment. And our goal is to elucidate the structures of these molecules, their detailed functions, and the mechanisms by which they are synthesized by the bacteria. On the application side, this group of metabolites has been responsible for over 50% of FDA-approved drugs in the past 40 years. And so developing methods to discover new natural products aids and facilitates drug discovery. Most of our naturally occurring cures have come from a small fraction of natural products that we can access under normal growth conditions in the lab. We can only capture maybe 10 or 20% of bacterial biosynthetic potential when we grow these bacteria in the lab. The majority of the biosynthetic pathways are said to be silent or cryptic. Our research is predicated upon trying to find ways to access this majority of secondary metabolites that we as a community have not been able to access in the past. We've developed methods that we call high-throughput elicitor screening, or HITES, that can turn on the production of these otherwise so-called cryptic metabolites that are not produced under standard growth conditions in the lab. And the way we do this is we tag a biosynthetic gene that we're interested in that's silent under normal conditions, such as green fluorescent protein. And this provides a rapid readout for the expression of that gene, which means if we do manage to turn it on, the bacteria turn fluorescent green, and so we can read that out relatively easily, relatively rapidly. And that allows us to screen many conditions to see just what it takes to activate these silent biosynthetic genes. Naturally, bacteria grow in a competitive environment with limited nutrients. And that's where their arsenal really comes in handy, because they can use it to outcompete other strains, to claim a niche, and to grow. If you contrast that to how we grow bacteria in the lab, in media that are very nutrient-rich, and importantly, where there are no competitors or predators. And so there's just no need to spend all that energy and synthesize half a dozen or more antibiotic or cytotoxic molecules. And so the way we grow them is just not conducive to capturing the full biosynthetic potential. That's exactly why the HITES approach works, because it creates an environment that bacteria are maybe more accustomed to, with sort of different types of molecules, perhaps toxic molecules that they have to fend off, and that elicits certain responses that they would probably generate in a natural environment. What we found is that low-dose antibiotics are actually the best stimulating molecules, the best activators of silent biosynthetic pathways. And using that concept, we've been able to isolate upwards of 50 new cryptic metabolites, and we're currently investigating these further with the ultimate goal of developing the best of these for clinical trials in the hopes that they can fortify our antibiotic arsenal against infectious disease. Tomorrow's cures, tomorrow's medicines might well be hidden in this group of metabolites, and I think HITES will be an important method in trying to tap into this new source of potential future drug.